السلام عليكم ورحمة الله وبركاته حبايبي، أخباركم إيه؟ إن شاء الله تكونوا بخير. كل سنة وانتم طيبين يا جماعة. معاكم طارق عباس، أستاذ البايوكيمستري والوراثة الجزيئية في جامعة فيرجينيا. النهاردة إن شاء الله هنبدأ أول درس بإذن الله في الصف الثالث الثانوي. زي ما وعدتكم إن شاء الله هحاول أقدم دروس مجانية على قناة اليوتيوب. ولو ما سمعتنيش في الفيديو اللي هو التمهيدي اللي بدأنا بيه وعرفتكم ازاي هنتواصل مع بعضينا، فيا ريت ترجع للفيديو ان شاء الله في القناه. رجاء الاشتراك في القناه وتشير الفيديو لزملائكم عشان الكل يستفيد ان شاء الله. طيب، النهارده ان شاء الله هنتكلم عن السبورت والموفمنت ان بلانتس. طبعا انا ان شاء الله دوت اول مره انا هدي درس بالانجلش، بس مش كله انجلش، هيبقى فيه طبعا عربي. بس انا هحاول بقدر الامكان ان انا اقلل استخدام اللغه العربيه عشان ان شاء الله كلنا نستفيد من المساله ديت. طيب، ان شاء الله انا هلتزم بالمنهج المتقدم في كتاب الوزاره زي ما انتم شايفين اهوت. والحقيقه الكتاب يعني متوزع لكذا جزء. الحقيقه يونت 1 ويونت 2. في يونت 1 عندنا 4 chapters. كل شابتر بيتعرض لمساله مختلفه عن الشابتر الاخرى. second unit ان شاء الله هنتكلم عن الموليكولار بيولوجي. ده طبعا تخصصي انا بالاساس. والحقيقه الجزء دوت is one of the most difficult parts that faces students. فان شاء الله هنركز عليه بشكل عميق باذن الله، وان شاء الله كلنا نستفيد. طيب، عشان انا هلتزم بكتاب الوزاره ان شاء الله I'm going to try to take certain pictures from the textbook, and every time I'm going to do that I'm going to indicate that this is a picture from your school textbook. At the same time, if I do reserve to go on and using some material from outside of the textbook, then I'm going to give you the reference for it so that you know where is it coming from. Of course, you are welcome to use any of my material. It's going to be distributed freely, inshallah. I'm going to be uploading that lecture on Google Drive, and I'm going to send you a link on my group on Facebook, as I've discussed before. Okay, so let's begin. Bismillah ar-Rahman ar-Rahim. All right, so we'll focus on the very first unit. The very first unit will deal with the structure and function in living organisms. And your textbook wants you to understand certain key knowledge in that area. It's divided into many little subtitles. We'll go over each one of those as we go through the lesson. Just try to follow me up, right? So my advice is to read the textbook. The textbook is not that big. It's a small volume, 130, 150 pages. So you're welcome to go through it to begin with. And it's a good idea to have an idea about what will be covered in the class as a whole, so that you have a good idea about what you're getting into. All right, so like I said, today we will be talking about the structure, right? Structure and function in living organisms in general. And your textbook, even though it does get some knowledge about other organisms, but in general we'll be focusing on humans, right, and plants, right? So obviously there are very big differences between these two different organisms. But when we deal with each topic, just like the one that we're going to start with today, support and movement, we will handle them one at a time, right? If there are similarities, I will be talking about what these similarities are. If there are differences, I will be talking about these differences. But I'll try to stick to one living organism, right, at a time. Dig deeper into that organisms, and whenever there's a potential for differences with plant cells, I'm going to do refer to it, and I'm going to do exactly the opposite. So for example, today we'll talk about the structure and function of living organisms. We will start with focusing on plants. Okay, so we'll start with plants, and that's going to take up to 2 or 3 lectures, right? And then we'll shift gears and handle the same sort of system, right, but in the human body. All right. So what I want you to do, right, to take most advantage of what we will be discussing is to refresh your memory about what you covered in the first and second year. Particularly second year is very relevant to this topic of support and movement in plant. So I've taken some snapshots from some of the pages in your textbook So I want you in particular to focus on chapter 1 and chapter 2. And for example, in chapter 1, you will see that the idea of permeability, right, of the cell membrane is critical for giving support, overall support to the plant cell. So I want you to really go back, right, and read these few pages. Starts with page 10, right, and then all the way to different pages. You want to go back and refresh your memory about how water and minerals are being absorbed at the roots of these plants. I want you to very importantly focus on the concept of osmosis and how is that going to become important for providing support to the plant cell. I also want you to focus on the idea about how transport is being carried out in higher plants. So these are very few pages, right? You're going to go through them very quickly. You've just finished the school year a month back, and so these ideas and these concepts are still fresh in your mind. So I want you to refresh your mind before we dig deeper into the little bit organization. Obviously, your textbooks in year 1, year 2, year 3 takes different concepts and builds on them in the next few years. So there's nothing such as that, okay, I'm done with year 1, why you're taking me back? No. You cannot really understand what's in year 3 in biology without having a strong foundation in year 1 and year 2. So go back and read chapter 1 and read chapter 2, the concept of transport, for example, of minerals and gold. I want you to go back and study vessels within the xylem, for example. I want you to go back in chapter 2 and look at the factor responsible for the scent, right? And so on and so forth. So chapter 1 and chapter 2, these are important refreshing material, right? So that you can build strongly as we go through the support system in plant, chapter 1 and chapter 2. All right, so let's begin by another review of a very important concept, which is the structure of the plant cell as a whole. Right, so obviously the plant cell is made of different components. So let me just refresh your memory, right? So you begin with what we call the cell wall, right? The cell wall, just from the surface of it, is this thick layer, right, that provides protection and support, right? Support. So since we're dealing with support, right, we will talk about movement in the second lecture, but in the first lecture, we'll focus primarily on the support. Right? And your textbooks, right, is telling you that there are different methods and systems for support, right? And it's already given you the function of these different methods and systems, and it summarizes them in 2 different points. To maintain its shape, right? So that's a support element, right? Maintain its shape for protection and for protection. So here, it's telling you that some of the support systems actually play a role in providing immunity, right? Immunity, right? And you'll see this theme over and over, that different systems do things other than what they are particularly designed to do, right? So the cell wall, for example, is designed to protect and give a shape and support to the overall entity of the cell, but it also protects it, right, from invading microorganisms and the effect of certain insects, for example, right? So it does 2 different functions. One is in the support system, right? One is in the transport of material across that wall, and another in terms of providing protection. Okay, and so these methods, so now it's going back and it's giving you a little bit details, maybe physiological, or maybe structural. And in fact, this is how your book is dividing support. Support to living organisms in general, right? Not just in plant cells, but in general, is divided into 2 big categories. One is physiological support, right? And the second one is a structural support. You'll see this theme coming on and on as we go through the plant cell and then the animal cells. Of course, these 2 units are not necessarily separable. For example, some of the physiological support mechanisms and methods are completely dependent on the structural formulation of these systems. And so there will be back and forth between these two phenomena. Okay, so obviously physiological always refers to function, right? Function or functionality. Whereas structure, right, is basically the composition, the chemical and physical composition of a given entity. And so some of you are very excited in this last year. Some of them are a little bit scared because they do have in their mind that they do want to go to medical school, for example. So in medical school, you'll study both anatomy, right, and physiology, structure and function. And these are two different topics, and you'll study them in 2 different years, right? You'll study anatomy 1 and anatomy 2, 1 in year 1, 2 in year 2, and physiology 1 and physiology 2. So 1 deals with the structure, right? Of course, in medical school, you'll be dealing with the structure of the human body and its different components, both at a macro level as well as on a micro level. And the second part would be the physiological system, where you start dealing with different systems that are made of different structures, right? Organs and suborgans sometimes. For example, we'll deal about the circulatory system, right? So the circulatory system is a system that's made of structural components, but in physiology, you'll understand how the system as a whole function together, right? So now we understand keywords. We will be dealing with support. Support has 2 components. One is physiological, one is structural, and there is a back and forth between these 2 intermingling phenomena. So back to the cell wall. So the cell wall provides a structural component to the plant cell. It maintains its shape. It gives it its structural hierarchy, for example, and also gives it a protective component. Let's see the different parts of the support system within the cell, because we will focus on the cell and then we'll go from the cell to the actual tissues and the plant as a whole. So when it comes to the cell, like I said, there is different components and there's some simple definitions and function, and I'll try to highlight these one at a time. So for example, we'll be talking about the structure of the cell wall. So that's the rigid outer layer, right, made primarily of cellulose. So cellulose is this type of sugar that you've learned in year 1 that's made of a complex type of sugars, right? Many monomers of glucose linked together, right? In the monomers are basically, let me see if I can, we'll come back to this point in more details, but basically the sugar molecule are linked together by glycosidic linkage with the next sugar, right? In the case of glucose, it's basically, we'll come again, so don't worry about this right now, but basically it has the glucose monomers linked together, right? Whereas each one is in opposite orientation to the next one. And this structure repeats many, many times, giving you the polymer, right? I will try to highlight some of the key words that we're using. Some of them are prefixes, some of them are suffixes, and often come from either Latin or even more commonly from ancient Greek. So for example, poly, right? If you remember from year 1, it means many. Actually, that's not Latin, that's Greek. The Latin word for many is multus. But poly means many, right? Whereas monomer, right? So mono, right, means 1, again in Greek, right? Di or bi means 2, right? And then tri, 3. And that doesn't apply only to the word monomer or polymer, right? But many, many, many times you will see this coming over and over. So poly always mean many, mono always mean 1, di and tri, 2 and 3. And then you have also tetra, right? 4, penta, 5, hexa, right? So for example, penta, you have the pentose sugar, for example. That's going to be, we're going to come back to this word in the sugar ribose, for example, where we're going to be talking about a pentose sugar, right? Sugar that's made out of 5 carbon atoms. Hex, right, or hexa means 6, hepta means 7, and so on and so on and so forth, right? So these little prefixes, it's important that you understand their roots. It's important that you understand that we can exchange different words with these different roots so that we can have a meaningful terminology that will help us understand the scientific sentence. As I mentioned before, this is really, most of it comes from, like I said, Latin and Greek, but the scientific world has adapted these terminologies, and we're going to have to, in order for us to advance, we have to adapt that terminology. And so I will always try to use the correct terminology, and maybe even correct some of the terminology that most common people are using, even teachers and even some scientists, unfortunately. So we're going to have to start correcting these terminology, and I want you to start learning the correct way of talking about these different things, right? So back to cellulose. So it's made out of these monomers of glucose subunits that are linked together by glycosidic linkages, right, in a very long molecule that basically is the most abundant macromolecule on Earth, right? Actually, it makes more than 50% of the total mass of biomolecules. So that's a really very important molecule that gives a lot of protection to the cell wall, right? So that's why, of course, the cell wall is made out of many other macromolecules. For example, you have the pectin, for example. Again, we'll talk about that. And that's why it's important to refresh your memory back in year 1 and year 2's material. So you have pectin, you have lignin, right? So you have so many other material that we're going to be talking about at its time when it comes. But right now, I just want you to understand that the outer layer is a rigid layer, right? So the cell can expand, right, but up to a certain point. And that's important when we talk about support. It can extend and expand as the cell grow in size up to a certain limit. And that particular wall gives it that limit and gives it that rigidity. And again, it provides structural support, right? So your book doesn't, your textbook doesn't really come into any close definition for what are the different components of the structural component, but you've actually learned them. But we will talk about some of the things that are not in the textbook, and the examiner will actually examine you on some of your previous knowledge from previous classes in year 1 and year 2, that you already know that particular wall does actually provide support. It doesn't mention it explicitly in year 3, but it's a vital component of the support system. Okay, I hope you are with me. And if I do have tendency to speak a little fast. So if I am speaking too fast for you, that you can understand what I'm saying, ask me to slow down. That's okay. I understand that that you're living in Arabic-speaking culture, and you're probably not used to listening to English in full sentences. And so I'm actually going to also help you speak like a scientist. And that's why what I'm trying to do is to be very careful for the choice of words that I'm using as I construct my sentences as I speak to you. All right, so the next element is the cell membrane. So let's use our pen again. So the cell membrane, obviously. So obviously this cell wall, right, is only present in plant cells and other cells, but not in human cells. Human cells do not have walls, right? But plant cells do. Now, cell membrane is another organelle, if you will. So the cells obviously is made out of many organelles, and organelles stands for a small organ. So the cell as a whole is made out of small little organs. We call them organelles, right? So the nucleus, for example, the mitochondria, the vacuole, even the cell membrane is considered an organelle. Some people also refer to the cell wall as an organelle. The cell wall itself is not covered by a membrane. It's actually an entity made out of complex sugar primarily, right? But the cell membrane and many of the other organelles, right, are enclosed within a cell, a membrane. And the term membrane is very specific and is different than the word wall, right? So membrane in biology often refers to this bilayer. Okay, let's break this down. Bilayer, again, so that's Greek for two. 2 layers. 2 layers of what? Of phospholipids. Okay, let's break this down, okay? I want you, when you read science, that you pay attention to every word and every subword. What is it made of? When we say DNA, right? DNA, that's a word, everybody know what DNA is, right? But really, you really don't know what DNA is until you dig deeper, right? So DNA stands for deoxyribo, right? Nucleic acid. So it's an acid that has a ribose removed from which oxygen an oxygen atom, and it's nucleic because it was initially discovered in the nucleus. Just a deoxyribonucleic acid, right? So now you know what DNA stands for. You go and walk in the street and somebody tells you DNA. He just know that the DNA is what carries the genetic material on the very surface thing. But you, as a person who studied DNA, you need to know what the name implies. Where is it coming from? Or what does it mean, right? At the details level. So let's go back to the membranes. So membranes, right, which encloses many of the cellular organelles, like the cell membrane here, like here in the mitochondria, they're all enclosed by cell membrane, or a bilayer of cell membrane. Here is again the chloroplast, which you learned in plant cell, again not present in a human cell. The endoplasmic reticulum, also bilayer of phospholipids, Golgi apparatus, right? Even lysosomes, right? The digestive system of the cell, the compartment in which the cell can take all the toxic material and all the dying debris and break them down using certain types of enzymes, not present elsewhere in the cell, but they must be enclosed by a cell membrane, right? The cell membrane. So let's go back to the bilayer composition. All right, so the bilayer is made of phospholipid and phospholipids, right? So phospho, right, it's a phosphate group, and lipids. So these are lipids, right? Attached to them, a phosphor group. How is it represented in general? So, you have a phosphate head attached to a fatty acid, right? Fatty acid, phosphate, right? And this phospholipids is repeated many times, right, forming the membrane. And it's made as one layer and a second layer. And the second layer are in the opposite orientation, meaning that the fatty acid tail is on top and the phosphate head is at the bottom. Right? So now you have, and this one extends throughout the cell. So in general, so this part here is just like a little bit snippet from what we're looking at right here, right? And this one, obviously, the two layers, these two layers, right, are extending throughout the cell to enclose the entire cellular compartments, right? So two layers, one layer, two layer. Well, is this arrangement just happen haphazardly, just stochastically like this without any meaning? No, of course it has a meaning. The phosphate head, right, is typically a charged head, right? It's a head, part of the molecule that is water loving, right? In other words, it's hydrophilic. So hydro means water and philic means loving, right? So this part of the phospholipid is a water loving part, whereas the fatty acid, by definition, right, you take some fat and like butter, for example, or oil, and you try to mix it with water, they don't mix. Why they don't mix? Because they hate each other. They cannot come together. Lipids are not dissolvable in water, right? So this part of the fatty acid, this part of the phospholipid, this lipidy part, right, is hydrophobic, right? Hydrophobic, right? Hydro, water, phobic, heats, right? So this part hates water, this part loves water. Now, of course, we know that outside of the cell, right, there are water or water-like material. And this part inside the cell is water or water-like material, just like the cytoplasm, for example. And so naturally, the part that loves water will face the water side, and the part that hates water will face to the inside. Okay, so if you do take just some mixture of phospholipids and you put them in water, what will happen? In other words, is this phenomena a biological phenomena, or is it a physical phenomena or a chemical phenomena? That's an important question, right? You're going to have to understand that question to understand support and many, many other biological concepts. You take a bunch of phospholipids, right? And you just put them in water. They will spontaneously form this bilayer, just randomly, right? They will form actually vesicles like this. You can see them under the microscope. The vesicle actually will be made of the bilayer that exactly resemble the composition of this part, right? Or the composition of the membrane that encloses the Golgi apparatus, or the membrane that encloses. So all the membranes have the same sort of structure, phospholipids. The one thing that's maybe a little different from organelle to organelle, right, is what's included in that bilayer, right? So you have a bilayer, for example. So let's say this is the cell membrane, right? So cell membrane, right? And we'll have another membrane, for example, that belongs to the lysosome, right? So the lysosomal membrane. Are these two identical? No, they're not. They both are made out of phospholipids, right? The same phospholipid, right? Hydrophilic part, hydrophobic part, two layers. That is constant in every membrane that encloses each one of those organelles, just like the vacuoles. But the difference is in the integration of some protein components in this membrane versus in this membrane. Okay, let me give you an example. So cells respond to many different signals, right? And to respond to the signal in the cell membrane, you must have the receptor for that signal. The receptor for that signal. And that signal comes in many different flavors. It could be a signal from an ion, sodium, for example, or potassium, right? And so there must be a receptor for that, because sodium or potassium alone will not be able to penetrate the cell membrane. So God created a system where the specific receptor for each cell, animal or human, right, have the right receptors on their surface, right? So these will formulate some gatekeeper, if you will, just right. So you have a gate, right? You have a wall, and on that wall you have somebody that allow access to the inside of the cell, and somebody can even allow access for material to exit the cell. So these are receptors, right? So the receptors on the surface of the cell membrane are different. And these are made of protein, the second macromolecule that we are talking about. So we talked about 3 different macromolecules right now, right? We talked about cellulose. That's a complex sugar. So that is the most abundant macromolecules, saccharides, polysaccharides, right? We talked about lipids and the phospholipids. That's the second macromolecule. And now we're talking about proteins. These are proteins that are integrated on the cell membrane of the various different components, right? So we said that, for example, there are certain receptors that must receive the signal and allow that signal to enter into the cell, gatekeeper. The lysosomal membrane is different. The lysosome itself as an entity is designed to digest certain debris and waste and even invading toxic materials. So by definition, it must contain within its lumen or interior, it must contain enzymes that are specifically able to do that, right? Without these enzymes coming out of the vesicles, right? So these enzymes must be protected. Why? Because if they are not protected, they're going to start digesting things within the cell, right? So the lysosome as an entity, for example, contains what we call hydrolytic enzymes. So proteins that are specifically able to degrade macromolecules. It will degrade sugars, complex sugars. It will degrade proteins, proteases, right? It will degrade even nucleic acids. It will degrade lipids, lipases. So these enzymes, we in general collectively call them hydrolytic enzymes. These enzymes must only work in an acidic environment. And since the pH of a cell inside of the cell is about 7.2, slightly basic, it's actually neutral, but it's slightly basic, 7.2, the actual pH of the vesicle must be kept under 5. It's actually between 4 and 5. Why? Because the enzymes that are most important for the function of the vesicle can only work in this low pH. Right? They only activated by low pH. So to maintain the low pH, how does the membrane actually work? To maintain that low pH, right? Now we're talking about the lysosome. Of course, your book didn't talk about any of this, but again, I'm going to just try to refresh your memory about the combination because this will come over and over again, but it's a good idea to actually start learning it from now. So that particular membrane has, like I said, is this typical bilayer phospholipids, but on its surface, it has something different than the receptors that we talked about is in the case of the cell membrane, these gatekeepers. What they do have in the membrane actually are specific pumps, if you will, that specifically take proton ions and pump them inside. It takes protons and pump them inside using energy or ATP, right? So this is a little factory, if you will, that has specific molecules, proteins, different than the proteins on the cell membrane of the cell, right? Specifically designed to take up both protons and also chloride. These are the two most important ionic composition of the lysosome. Why? This is important to keep the pH here low. Why? To activate enzymes important for digestion. These enzymes would not work except within the lysosome. So God created a system, right, very similar to the stomach, where the digestive part of it is hydrochloric acid, where you can digest food, for example, right? Break down these macromolecules so that the body can take advantage of the material. In the lysosome, this is the little digestive system of the cell. In fact, people think that the vacuole, right, which is probably the largest organelle in a plant cell, is pretty much a large lysosome that also have the function of storing food, right? Excess food for storage, sugar, right? Minerals, salts, but also has some of the enzyme that can actually break down these things. There's a big contradiction or controversy in terms of whether the vacuole is a true lysosome or not. I'm not going to get into that. But again, so the point that I'm trying to say is that every organelle carries out specific function, right? And their cell membrane is designed to allow the entry and the exit of a specific molecules, very specific to that compartment. I can go on and on and on, but I think I beat that point, right? So we have a cell membrane that is semi-permeable. Why did we say the word semi? And that can become very important for support, right? Why did we say semi? Because it does allow the movement of water, for example, but it doesn't allow the movement of protons without the pump, right? So without the pump. So if you don't have the pump, this membrane allows entry of water or exit of water, but it doesn't allow the entry or exit of other bigger material or other different material. It's selective for that function. So these membranes are selective. That's the other word I want you to understand. If I am a good gatekeeper right here, standing on a particular gate for entry, let's say at the airport, right? You come in, you can pass. You're not allowed. You've got a gun in your pocket. Go somewhere else, right? So I'm being selective. I let somebody come in and I prevent other from coming in. And there's another gatekeeper doing exactly the opposite. I allow something to come out or prevent somebody from coming out. So these membranes are semi-permeable membranes, right? They're composed of phospholipid bilayer, like I said, right, with embedded proteins. And the different composition of these proteins is what gives each membrane its particular characteristics. Providing a barrier, right? Immunity. That security guard that's standing on the wall, that provides immunity. It prevents the entry or exit from other things. Another function. So when you come to the exam questions, right, the examiner wants you to think, okay, the cell membrane or the cell wall, let's focus on the cell membrane. Does it play a supportive role in the cell? Sure. How so? It allows entry for the water. It allows entry for the water. The water will build up in the cells, will allow the cell to expand and extend, and gives it its rigidity and structure, right? The water must enter and collectively enter into these little vacuoles, and it will push the vacuoles out, just like you're blowing an air in a balloon, right? Or in a soccer ball, right? You don't have that air. You fill it with air, it expands, expands, expands up to a limit. But once it's expanded, it's rigid, it's tough, it's standing on its feet. The plant is upright, right? So the cell membrane then, by definition, provides support. Only, so if one of the answers in the question, only, the answer is no. It's not only, right? That's not the only function of the membrane. Of course, the membrane, is it physiological support or is it structural support? Well, if you focus on the fact that the cell membrane allows entry of water, and that by the process of osmosis that we will talk about in a second, and that provides physiological support, it is also providing a structural support as well, right? It's also providing a structural support. How so? Well, the structure of the phospholipid itself, the proteins that allow the different ones. So there's a little bit of trick here, right? That what's the major function of the membrane in support? So then the person that puts the exam must be very careful when it formulates the question, right? Does it provide physiological support? And if it asks you only, then you have to think, does that particular organ or organelle play a different role in addition to its role in support? So you have to be very careful for the wording of the question, right? I want you to think about what the question is really saying and what's really asking. Of course, sometimes the examiner used the wrong formulation of the question, but by exclusion, right, of the other answers. And again, I want you to, whenever you face with multiple choice question, is to actually read every single answer, even if you know the answer from the beginning. But sometimes one keyword in these little answers actually tells you, no, this is not the right answer. So not only that you need to read and carefully understand the question, but I also want you to read and understand each of the answers, so that when you have a question where, for example, this one provides support only structurally, or also something else, or just leaves it open for you, I want you to think about what you understand as a student at this stage about the different functions of the different part. And that's why I'm digging deeper into these little elements that are completely outside of your notebook, out of your textbook. Of course, the examiner allows you, with this new system of examination in high school in Egypt, the examiner or the Ministry of Education wants you to start thinking more than just memorizing what's in the book. And I think that's the right way to do it. It's a different system than what you're accustomed to, but you're going to have to start adapting that system, because actually advances in the sciences require that sort of type of learning. Okay, so we're too slow, so we're going to have to push it up a little bit. So let's see what else are here. This is one of the reasons why I said, okay, go back and study the structure of the plant cell. Go back and study how water comes in and out. Go back and study how minerals and ions are transported within the plant cell. I think that will help you understand the 2-3 sentences that are provided in your textbook. Okay, cytoplasm, obviously this is a jelly-like substance. It's water-like, mostly made out of water. Salt and proteins, all of you know that, right? The word cytoplasm sometimes come in plasma cells, sometimes most people actually say the protoplasm. Actually, there's a difference in what cytoplasm is and what protoplasm is. Cytoplasm is everything within the cell wall, right, except the nucleus, whereas the protoplasm includes the nucleus as well. So, I don't think your book is actually making that distinction, but if you see the word proto, right, proto means primary, plasm, you all know what plasm is, jelly-like substance, right? Then you know that they're pretty much exchangeable, and most people erroneously use that exchangeable terminology. But we'll stick with the word protoplasm, but if somebody says cytoplasm in a plant cell, then you know what he's talking about, or she's talking about. So cytoplasm excludes the nucleus and organelle within it, the nucleolus, whereas protoplasm includes the entire thing. All right, so of course the cytoplasm also is important because it contains all these materials. Cytoplasm actually provides the material, the media, right? The media, the watery media that forces the formation of these vesicles. Without the cytoplasm, you're not going to be able to form any of these vesicles, just like we described in the bilayer composition in each of the membranes that encloses each of the cell organelles. The nucleus, this is the housekeeper for the genetic material. The second part of your textbook is dedicated to studying the genetic material, DNA in particular, but also will expose you to transcription and RNA, as well as protein buildup. We're going to talk about that in much, much greater detail, obviously, and then when we come to that point. But for now, I just want you to know that, For now, I just want you to know that the nucleus harbor our chromosomes, right? In the case of plant, it harbors the genetic material in the form of chromosomes, and obviously different plant cells have different amount of different chromosomes. You're not expected to know any of that, but just know that this is where the DNA is condensed together. This is where each segment of that DNA represented as genes will then function as a blueprint from which the genetic trait will be expressed to give us life, right? So this is the source of life, the genetic material made out of deoxyribonucleic acids. So here's a nucleus. So this is a large, again, membrane, right? Single membrane bound organelle. Its function acts as a control center. This is where gene expression, right? Certain genes will be expressed in this plant cells. Other plant cell will express different set of genes. All the expression begins from the DNA, right? And then, of course, the mRNA will exit and then gets expressed into a protein. Again, we'll talk about that in details in the second part, and so don't. Right now, I want you just to refresh your memory about the different components and what their main functions are. We haven't started talking about support yet. I'm touching to them every time that I'm discussing one of those points, but we haven't actually dealt with the topic at hand. Again, this is the first lecture, so be patient. I hope you're enjoying the lecture. I hope that you're having a good, refreshing understanding for the different composition of the different parts of the cell. Okay. Of course, we have also nucleolus, right? This is where the ribosomes are assembled. This is where the machineries that are important for translating RNA into proteins are built up, and then they're exported to the cytoplasm where they carry out the topic of translation. Again, we'll talk about that in more details. There was actually one question in this year's exam for the high school grade 3 that was very controversial about the different functions of the nucleus or the nucleolus for carrying out a specific function. And when we discuss that section, we'll have fun. Okay, we also have ribosomes. Again, so I just already said that this is where they're made right here, but ribosomes are also present, right, on the surface of the endoplasmic reticulum, forming what we call polyribosomes, right? Many ribosomes, but also there are free ribosomes throughout the cytoplasm as well. So the ribosome is basically the small particles, and that's basically not considered an organ. It's considered an organelle, but actually the ribosome is just a multi-subunit protein. It's a huge complex of many proteins and certain types of nucleic acids, but without that membrane that I've been talking about that encloses every other material. So this is one of the very few membraneless, right? Okay, so you guys are studying science in English, so now understand here's a suffix, right? Membrane less, right? One word, right? But break it down into membrane less without membrane, right? So a membrane less, so I can add to this here, right? Membrane less small particles, right? Composed of RNA and proteins. They're found either freely floating, just like I said, right? Or on the surface of the endoplasmic reticulum, the part that we say the rough part of the endoplasmic reticulum. So obviously the endoplasmic reticulum here is the rough part. You call it rough because their surface is filled with these polyribosomes, whereas the smooth endoplasmic reticulum lacking these polysomes. So ribosomes themselves are the sites of protein synthesis. They are assembled in the nucleolus. They exit the nucleolus into the nucleus and ultimately to the cytoplasm, where they're deposited on the surface of the rough endoplasmic reticulum, where they carry out protein synthesis, translation and synthesis. The ER, right? So these are, again, so this is one of the biggest organelles in the cell. It's a network of single bilayer, phospholipid bilayer of membranes embedded with certain proteins, different than those on the cell surface, different on those in the lysosomes. Basically their main function depends on whether they're rough. If they're rough, the main function obviously is the ribosome function, which is synthesis of proteins and also processing of these proteins, adding or taking away certain elements from these proteins. Whereas the smooth one, right, they lack ribosomes, like I said, and their main function is to synthesize lipids as well as to detoxify, remove. D, right? D means to remove, right? Certain toxins, so detoxify certain chemicals. Okay, what else? Okay, we have the Golgi apparatus. We already exposed to that. So this is a stack, membrane-bound vesicles. Again, membrane bilayer phospholipid, different proteins integrated on their surface. And their main function is to modify, sort, and package different proteins and lipids for storage, right? They're also an important part of the transport system of the cell, right? That's different than the transport system outside of the cell that you're going to be refreshing your memory on from chapter 2 in year 2. What else? Vacuoles. So your textbook talks about support, talks about physiological support implant, and the textbook wants you to know that this particular organelle, right, enclosed within a single membrane bilayer made out of phospholipids, is the most important, your textbook suggests, physiological support system for the plant cell. Why? Because the water comes from outside the cell to the inside of the cell, right? And expand these vacuoles. Obviously, this is a picture for only containing one vacuole. This is a large vesicle, but many plant cells have multiple vacuoles. And as these get filled with water, then they start pushing on the cell membrane, extend these membranes, extend the cell, make the cell stand alone in a tough way. And that's building what we call the Turgor pressure, or ضغط الامتلاء بالعربي. We're going to come to this in a second. And that's what keeps the cell inflated, right? Tough, just like a very full-blown soccer ball. So you can actually kick it and score a goal with it. All right, so it's a large membrane-bound sac, right? Or a bag, or a vesicle filled with fluid, mostly cytoplasmic fluid, right? And its main function is to store nutrients, as I suggested. It also has some certain waste products, and it helps maintain the turgor. That's your key word here. Okay, so every time that I'm going to be underlining something, every time I'm going to be underlining something, I'm going to basically, by underlining and by highlighting, that means that this is a key word that the chapter is emphasizing that's relevant to the topic at hand. In our case, the support part, right? The support part. So the turgor pressure that contribute to the cell growth by absorbing water, right? And giving physiological support. Why we say physiological? Because it depends on the ability of these membranes to suck up water, right? From outside, from also inside, right? Through the process of osmosis. And osmosis, right, is not a structural component. Osmosis is a physiological phenomenon. It's a physical phenomenon, right? That's dependent on the concentration difference of different solutes within an environment, between two different environments. We'll talk about this in greater detail. So that's what it is, right? So that's basically provides physiological support, primarily, okay? Is it only? No, it's not only, but primarily physiological support. Why? Because it's the ability to absorb water and build turgor pressure. Okay. Chloroplasts, again, there's nothing much to add to what you already know about chloroplasts. This is the double membrane organelle. This is a little bit different organelle than the other single membrane organelle. So all the membranes that I talked about before were actually single membrane, but is made out of two layers, bilayer, but it's a single membrane. Membrane includes the two layers. The chloroplast is a double membrane, right? Each one of them is a bilayer. So that's a double membrane enclosed organelle, right? And obviously this is only unique to plant. Animal cells do not have chloroplasts. Some fungi have chloroplasts, but animal cells in general don't have chloroplasts. Their function, obviously, is to photosynthesis, right? Which we'll come to in a different time as we move forward in the class. Obviously, it converts light energy into a chemical energy stored in sugars, like glucose. Mitochondria, again, another second double-stranded membrane. People actually think that both mitochondria and chloroplasts originated from one another. They're very similar in function, except that mitochondria cannot perform photosynthesis, whereas chloroplast does. So chloroplast also is the energy production factory, right? So it's a double membrane organelle. You can see it right here, right? And it has what we call different inner folding. They call them cristae. So these little folding here. Function, produce ATP through cellular respiration, right? So we, as animals, we get our energy from respiration, right? Plants get their energy through the sunlight, converting sunlight, using two very similar organisms. Cytoskeleton. Your textbook doesn't talk by any means about the role of cytoskeleton in support, right? But in fact, the cell cannot maintain its integrity without the functionality of the structural component of the plant. So this is a structure. It's made out of network. It's made out of network, right, of protein fibers, including microtubules, microfilaments, and intermediate filaments, right? So this is certain type of proteins, certain types of proteins, certain types of proteins, right? Inside the cells, if I just take away any of all these organelles, the actual integration, right, within the cell, there's this in a very elaborate system made out of the cytoskeleton, right? Cell, right? And skeleton, هيكل, right? الهيكل الخلوي, right? You cannot have support without having a skeleton. In fact, when the book deals with the human body and talks about support, it's entirely dedicated about the skeleton, right? the cranium, the spinal spines, right? Vertebra, the hip bone, right? The skeleton of the cell, right? The skeleton of the cell is what provides the structural support for the plant cell, right? One of the most important proteins in that cytoskeleton is actually what we call actin, right? As we talk about the second or third chapter in the book, we'll deal about the muscle cell, right? The muscle cell is made out of muscle fibers. Obviously, the main function of the muscle is to contract and relax. And so that function is made of two proteins. One is called actin. The second one is called myosin. This is a structural protein. This is a motor protein. They do come together, right, and provide support. We'll dedicate a whole lecture or two on that alone. But actin is also a component of the cytoskeleton, right? Similar to the microtubules, similar to the filaments, element 2, that particular. So this is a structural component, right, of the support system in a plant cell and in every cell, plant, animal, fungus, bacteria. They all have cytoskeleton. All right. There's another part of the plant cell that the book doesn't talk about, but that's not very important for you to know. That's why I highlighted in blue, which talks about the plasmodesmata and that particular organ. This is just a channel between the plant cell walls, right? So if I have a second cell here, right, or here, or here, so these will be channels between the different cells so that it allows the exchange of material, nutrients, and what have you, as well as communication between adjacent or neighboring plant cells. Okay, so with the end of this particular one, obviously there are many other components of the plant cell that I'm not going to talk about, but I just wanted to highlight and refresh your memory about what the different structures. And from that, we already have taken our information, right? We have a vacuole that's important for the physiological support. We have a cell membrane that's important for both physiological, because it allows water and solutes to come in and out, as well as structural component. We did talk about the cytoskeleton, providing a support system, an internal support system that allow for the cell to maintain its integrity and rigidity when it's well nourished. Okay, so now we begin with your book. Okay, so this part was an introductory part, and now we will shift to actually dig into the textbook. So the textbook wants you to know about support and movement in plant to these 3 sections, taken from your school book, right? So again, let's see what they're saying. So it's defining for you what support in plant, and we said these are system and methods, right, that are important for shape and protection. Shape and protection, right? And these methods could be physiological, right, or structural. Structural, like the cellulose, right, the most common biomolecule on Earth, the lignin, right? Very similar to cellulose in composition. In fact, when you eat vegetables, for example, the fibrous part that's not digestible, that's what the lignin is, right? So these are green fibers. If you chew on any particular green plant and you're not able to digest it, these fibers that we call, right, they're made of the sugar lignin, very complex sugar, similar to cellulose. Okay, so these provide structural support, and they're also physiological support. Your book wants you to know that this, most importantly, this process of osmosis, right? And these are the other parts. So physiological supports, we can start reading it. If you soak dried fruit and water, right? So dried fruit, right, in water, you will notice on time that the plant will absorb the water and become nicely inflated while maintaining shape. And it gives the example of a split bees, right? Very well nourished. If you just have it like that, if you take it out of its environment and leave it for a while, it will dry and will start shrinking, right? Same thing for the split bee. So the split bees, and same thing for beans. I gave you the other example, beans, right? So it basically wants you to understand that the main idea here is that water can get into the well-nourished plant and gives it its shape and its skeleton, whereas if you don't have that characteristic, this osmotic pressure allowing the water to come in, right, then the plant will shrink and lose its support, right? Loses swelling and tension. So we can say that the cell become turgid, right? Remember, turgor pressure, the cell is turgid. When the water passes by osmosis, right? I did talk about this in a second, and I'm going to elaborate on this more, right? So as a result, the size and the pressure, and again here specifies for you, right? The word vacuoles, right? Turgid. See, I'm giving you the keywords here. You're going to be examined on these keywords. So I want you to pay attention to them, right? Passes by osmosis, right? Into, right? Its pressure increases, which blah, blah, blah. As well as wilting of leaves and stems. So basically giving you that the turgidity, right? Turgidity, same thing. Wilting, right? So keywords, right? Very small sentence talk about physiology of the support system in plant. Obviously, this is very limited vision, but we'll stick with that. And then the structural component, which will be covered in the second lecture. I'll leave it there, but I already hinted to it, right? The formulation of these cell wall made primarily of cellulose, lignin, and other material is basically what provides the structural support of the plant. We did talk about the cytoskeleton and other material not mentioned here, but it gives you some keywords so that you understand what's happening here, right? And then we'll be talking about these things in bigger details, but that's where you need to go back and revise chapter 2 in your textbook. All right. We already covered this. Osmosis, right? So this is the first part that talks that your textbook is talking about the importance of osmosis. So, excuse me. So what I'm going to try to do in the next sentence or two is to highlight what is osmosis. So, and I'm highlighting to you, right, in red, the keywords, right? So one is, let's define what osmosis means. Osmosis, right, is a net movement of water molecules across a semi-permeable membrane. So let's use our pen again, right? So keyword here is the net movement. What does that mean? The net movement. So if I'm standing right here in the middle, right, and somebody is pushing me from this side, okay, and somebody else is pushing me from this side, will I be moving if they're both equally forceful? One will push from this side, one will push from this side. I'm standing still, right? I cannot move. But if this guy that's pushing me here is giving me more force to push me this way, right, I'm going to move to this direction. This little net movement is what I refer to here by net, right? You go to the market, you buy something, you give that person $10, right? That thing that you bought is worth, or whatever currency you're using, right? I said $10. They'll take $5 from you, right? Give you 5 back. What's the net difference? The net difference is $5, right? 10 minus 5 is 5. So osmosis, right, refers to the net movement of water molecule. How much water molecule will move from the outside to the inside? Obviously, water comes in and out, right? If the cell is not well irrigated, water will be lost. If there's more water outside, it will come inside. What drives this movement of water inside and outside the given enclosed system is actually the concentration, right, of the solutes within that environment on the inside versus on the outside. That difference was important. And so here is a net movement of molecules across a membrane. The membrane must be permeable. It must allow the water to come in and out, right? And there must be a membrane, right? There must be a division where the more water would move from one direction to the other direction. So you have two different environments separated by, so environment 1, environment 2. They are separated by a semi-permeable membrane. And the water must be able to move in one direction versus another. The question is, what drives that movement? Right? What drives the movement, right, is the difference in solute concentration. You've studied this already in year 2, right? So if there's more sugar, for example, on this side than in this side, what will happen? The sugar, naturally, if there is no membrane, right? If there's no membrane, no separation, the sugar will simply diffuse. Diffuse. You heard that word before? Yes, you have. Diffuse, right? Diffusion. The concentrated solutes on one given side, if there is no barrier, will simply diffuse so that at the end of the day, right, there is equal concentration. If you have 10, let's say, molecules of sugar, they'll be equally distributed throughout that volume. They'll diffuse from the high concentrated area to the low concentrated area simply by diffusion. Here, the sugar molecule cannot move because the membrane is impermeable to that solute, right? What will happen? Instead of the molecules go in, the water will come in, right? Now, by having the water coming in, it allows for the expansion of these well-concentrated ones, right? So that net movement is then driven by the higher concentration of the solute in one environment versus that in the environment within which the water, that same solute is at low concentration. Let's take the example of a plant cell. If the plant cell is buried in dirt, for example, right? If the concentration of the sugars, right, the sugars and the minerals are much higher here than a malnourished environment, there's no nourishment, there's no water, it's a dry environment, what will happen? You have much higher concentration here. Now you put some water somewhere outside, of course the water will come in, right? Why doesn't the water come out? Because the water come in from the environment when there is low concentration of solutes to the concentrated solutes within the cell. So that's the process of osmosis, right? So it's a net movement of water, not the solutes, but the solvent, right, across that semi-permeable membrane. So you must have the condition of the presence of a membrane. This is why I spent half an hour on describing to you what is the membrane made of. Somebody will challenge me, okay, why am I giving you all this information? No, you need to understand what the word membrane means. You need to know why this membrane is semi-permeable, right? Why does it allow the entry of water, right, but not the entry of other chemicals, complex chemicals? Why are there some gatekeeper that allow for the entry and exit of certain molecules but not others? They all control the permeability of the membrane, allowing certain things to come in and certain things to come out. So our membranes, cell membrane, not the wall, our cell membrane is semi-permeable. What about the cell wall? Is it permeable? It's much thicker than the cell membrane. Is it permeable? Yes, it is. It's freely permeable. What does that mean? It means that water can come in and out this area, no problem. What it means also, it means that any other chemical, like maybe some free sugars here and here, they can exchange nicely and freely by simple diffusion, right? If you have some water here and this is completely filled, then this layer becomes saturated by water, just like a sponge. Okay, so we have a process of diffusion, very different from osmosis. Osmosis requires the presence of a permeable membrane and a difference in the ion concentration. So what if I have equal amount of solutes on this side, similar to this side? Will be there a net movement of water? There will be movement of water, but it will not be a net movement of water, meaning that water will come in and will go out freely, right? But there is not going to be a net gain or a net loss. They are at balance. They are at equilibrium. Okay, so that's osmosis. So osmosis is dependent entirely on the presence of a semi-permeable membrane, on a difference on the concentration of solutes on either side of this membrane, right? And that membrane must be permeable to water. Otherwise, water will not enter, right? Must be permeable or bilayer, but impermeable to the solute molecule. Okay, so now we know what mitosis is, what osmosis is. Mitosis, sorry, meiosis. Osmosis. Okay, let's continue our discussion of osmosis. Here is another description of what I just stated. So you have a semi-permeable membrane. This is similar to our plasma membrane, for example. And you have one side of that membrane has many more concentrated substance, sugar, proteins, lipid, and I'm sorry, not lipids. Lipids are not soluble in water, but sugar, proteins, nucleic acids, any of those things, right? So this could be sugar, it could be salt, it could be minerals, it could be ions, right? It could be proteins, right? Many macromolecules. So long as they are soluble in this environment, they're all considered to be called solutes, right? So if there's more concentration of these substances on one side of that membrane versus the other side, then osmotic pressure will pull the water into the direction of the highly concentrated substance, right? The membrane must be right? Semi-permeable. Not permeable, semi-permeable. If it's freely permeable, right, then these solutes will move by diffusion, not by osmosis. Okay, here's a more comprehensive definition for those of you that like to understand things in much more comprehensive details. So it's a process by which water molecules move across a semi-permeable membrane from an area of low solute concentration to an area of higher solute concentration, which is exactly what I stated before. And this movement aims to equalize the solute concentrations across the two sides of that membrane. And that's a very fundamental concept in biology and plays a crucial role in maintaining cell homeostasis, right? Cell homeostasis, the physiological support system that pushes on these vacuoles and expand and extend the cells to its limits so that it maintains its rigidity. Okay, so beside this osmotic pressure that we talked about, we need to also understand what we mean when we say the turgid pressure, right? So turgid pressure is very similar to the osmotic pressure that pushes the water out, but as long as you keep building water inside the cell, right, this is not going to continue to have to expand anymore. So now you have reached a point where the cell is no longer extendable, right? You already pushed it to the limit. You already filled the balloon with air all the way to the end. You push it anymore, that balloon will explode. Of course, that's not going to happen if you keep inflating something with a very rigid wall, right? It's not going to explode in a second, right? And what prevents it from exploding is the buildup of that, what we call the turgor pressure, right? So the turgor pressure is now you already have filled that side with enough water, right? So now the difference in that water here versus there is now putting a different hydrostatic pressure on the opposite side. It opposes that osmotic pressure, right? In Arabic, we call it daft al-imtila, right? You already filled it with water. You already have built up enough pressure inside that compartment that now pushes against the tendency for water molecules to move across that brain, right? So this develops inside the plant cell as a result of water uptake by osmosis. It is also the driving force for cell expansion in plant growth and maintains the rigidity of the plant plant stems and leaves. Okay, so I want you to understand what are the conditions that are needed for building up an osmotic pressure. We need a membrane, semi-permeable, allow some but not others to go across. Difference in the concentration of solutes between the two different compartments. And this is opposed by a hydrostatic pressure that builds up in the cell that maintains that rigidity. Okay, so let's give an example. Obviously, osmosis is not something limited to a plant cell. Any cell, you put it into a concentration, a solution made of, let's say, water, and you change the concentration of the solute within that water, the cell can respond in a different way depending on the difference of the concentration of that solutes inside that cell versus outside the cell. So here's an example. You take a red blood cell And the question actually is, even though we're discussing plant, but it's very relevant to an animal cell. And you can actually have a question about osmosis, not necessarily in plant cell, because again, the examiner wants you to think about the phenomena more so than is it only applicable here, but does it also extend outside of the plant cell? So it may give you the idea, okay, what happens if you take a red blood cell and you put it in what we call an isotonic solution? Isotonic solution means a solution in which the concentration of a given solute, right, is equivalent to the concentration of that solute within the cell. So that's called an isotonic solution. What happens is the cells maintain its shape. So for example, the blood, right, the fluid blood in which the red blood cells are circulating normally is an isotonic solution, is a solution in which the amount of solutes within the actual liquid part of the blood is similar to that inside the red blood cells. So what happens if you now have a person that has, let's say, much higher concentration of, let's say, salt in their blood. Let's say sugar, maybe that's more common, right? A person that has higher concentration of sugar in their blood, right? A hypertonic, right? Higher concentration of sugar in the blood. What will happen is that water will come from the inside of the blood cells to the outside into the blood by osmosis, and the cell will shrink, eventually die. Same thing, you take a plant cell, you put it in a hypertonic solution, a solution in which the concentration of a given solute is higher than that which is in the cell, then those plant cells will shrink, will crenate. If you do the opposite, right, you take away sugars, right, or you put the animal red blood cells in a hypotonic solution, a solution in which the concentration of that solute is lower, right? What happened? You have a much higher concentration of solutes inside the cell. Water gets into the cell by osmosis. Why by osmosis? Because the animal cell is covered by a semi-permeable membrane, and now you have two environments, inside and outside the cell membrane, that have difference in the solution, in the solute concentration. Of course, there will be some question and answer time where we're going to give some examples into, for example, a solution concentration made out of, let's say, 30% sodium chloride, right? What does that really mean? Or versus 40%. We'll discuss that in much more detail. Now, you increase that hypotonicity, right? Very hypotonic solution. Then, of course, you're pushing that more and more, right? You keep inflating that balloon with water, and the cell will lyse, rupture. And you could then extend this to any living cell or any non-living environment in which there is a semi-permeable membrane. And there are many applications that drives from this buildup of osmotic pressure that depends on the presence of a semi-permeable membrane. All right. So the importance of this tilt trigger is in plant is basically osmosis is very critical, right? Again, the textbook wants you to know that this is the most important physiological function, right, of the support system in plants, is this use of osmotic pressure to maintain rigidity of the cells, but I've already mentioned several other ones. This second importance of the osmosis is basically osmosis have two main functions, right? One is to provide the cell turgor, right? This rigidity of the cell, this build-up pressure that result from the uptake of water into the cell. And second is regulation of internal environment, because osmosis helps regulate the internal environment of cells by controlling the flow of water in and out of the cell, maintaining cell volume and function. And that's important, and that also plays a role in the actual movement of fluid inside the cell. We'll deal about that when we talk about the movement component, right, of our lecture, support and movement in plant. We haven't touched that yet. The third function basically is nutrient absorption and waste removal, because again, that process is dependent on the difference in the amount of the concentration. So osmosis facilitates the absorption of nutrients and the removal of waste product in various biological systems. We didn't talk about that much, and I don't think you need to worry about that part. I want you to think about the word when I say, okay, that osmosis, right, and rigidity, it doesn't have to apply only to living cells. And so some of the question that may come to you in the exam, okay, well, if you take a dead cell or a dead plant and you put it in water, will it take up water by osmosis, right? Of course, to a certain limit, right? Because it's still a living cell. So you have a living cell, right, that has a membrane. This cell, living cell, will take up water by osmosis, just like what we described. If the cell died and still maintained that cell membrane integrity, it will take up water by osmosis. But when the cell die completely out, right, you no longer have that semi-permeable membrane. So it's not whether the cell is dead or alive, it's how long did it pass after the cell died. If the cell died but the membrane is still intact, sure, cells will take water by osmosis. For example, if you are a gardener and you take some flowers out of its roots, right? You leave it outside a little bit, they'll dry out, and so on and so forth. The cells have maybe started to die. You give them some water, they'll actually build up that turgor pressure and they'll look fresh and still young and alive. After certain points where the membrane started to break down, when the membranes break down, you add no matter how much water you put, nothing's going to happen. Why? Because you've lost that condition that we talked about, that semi-permeability of the membrane. The cell already have sterilizing their membranes. You no longer have that barrier. There's no such a thing as now you have two different compartments. One has lower and one has higher concentration. And so now we're talking about complete death of the cell with the dissolution of these membranes and no longer having to have that. So be careful with the wording of the question. Does he imply freshly dead or dead long ago, right? Such that you no longer have the condition, because the answer will depend on the integrity of that semi-permeable membrane. Okay, so how do we calculate osmotic pressure? Osmotic pressure is a term, it's a physiological term, it's a physiochemical characteristic, right? And we refer to it by the sign pi. Actually, this calculation was discovered by this great scientist, this Dutch great scientist. His name is Van 't Hoff. This guy, born in the Netherlands and died at the age of 58 in Berlin. He actually studied in Berlin and lived in Berlin, Germany. And he was the very first recipient of the very first Nobel Prize in Chemistry in 1904. Great scientist. Of course, not mentioned in your book. You're not going to find this question in your book. Sorry, this equation in your book, but I think it's really important to talk about osmosis. How are you going to calculate it, right? And many questions could be driven by that. So I want you to understand how actually osmotic pressure is calculated. And again, it was discovered by this great scientist. He put us an equation. He actually used to be working first on gases. He is a scientist working in the field of physical chemistry. And he spent a great amount of his career studying the behavior of gases. Later on, he switched to the behavior of fluids. And the question of osmosis was something that was bothering him for quite some time, and he decided to understand it better, and he came up to us with an equation that allows us to actually be able to accurately calculate osmotic pressure. Let's go over this formula. I don't want you to memorize this, okay? I want you to understand the main meaning of each of these things, because you're not going to understand osmotic pressure or osmosis until you understand the various components. So his formula basically is pi equal IMRT, right? And each one of those refers to something. Pi is, again, the osmotic pressure. Pressure is always measured in different things, and we'll talk about that in a second, but basically atmosphere. Of course, to liquids, we often talk about pressure in terms of like millimeter mercury, but in gases, typically atmosphere is the right unit that people use. I is Van Hoff. It carries the scientist's name, right? This great guy, scientist's name. So that's a factor that refers to the number of particles of solutes that are being dissociated into it, right? So here it is. So you already have a relationship between the osmotic pressure, right, and the number of particles, right? As you increase the number of particles, you increase, of course, here's your membrane, right? As you increase the number of particles that are dissolved in a given solution, the more the osmotic pull-up, the pull pressure that you have. The more I, the more pi, right? Second factor is the molar concentration, right? How many of the different solutes, right, are present in the water, right? How many, not dissolved, but how many molecules, the concentration now. Now we're talking about concentration, right? Concentration is how many per what volume, right? How many of these molecules by what volume? You cannot tell me that when I saw concentration is just 50%. No, that's just not enough. You have to tell me how many grams, how much weight, how many molecules, numbers in a given volume. You don't just give me the number of weight. You don't just tell me 5 grams. No. 5 grams in what volume? Because 5 grams in 1 liter is not the same thing as 5 grams in 10 liters, right? So when you refer to the word concentration, make sure you use the correct units, right? So the molar concentration, whenever we talk about salts or what have you, material like chemicals that we use in biology, it's always in like molarity, right? So the molar mass, right, of the solution, the more the concentration, right, the amount of the solutes per a given volume, how much volume do I have, right? The more the mass, right, within a given volume, the more the pressure you have. So if you have 2 different compartments, one has less concentration here versus nothing, and another, but more concentration here, right? So this is more concentrated, this is less concentrated. Is the osmotic pressure here the same as here? Of course not. Osmotic pressure here is much larger. Why? Because there's a direct relationship between osmotic pressure and the molar mass that we have, right? And then the third item, R, is the ideal gas constant. This is just a constant that from his calculation came up with. And if you've taken physics right now in year 1 and 2, and so you understand that most of the physical phenomena require a given constant for them to solve their equations. And in this case, we have an ideal gas constant that has a certain size. And then finally, and perhaps most importantly, is temperature in Kelvin, right? The osmotic pressure varies with temperature, right? Again, the relationship is a direct relationship, meaning that as you increase the temperature, right, you increase the tendency for the osmotic pressure to also become larger. Why is that? Because when you have solutes between 2 different compartments separated by a semi-permeable membrane, and you have given solutes on one side versus another, right? As you heat that environment, what will happen is that these molecules move, right? The ions move very quickly. And so that builds up more pressure here and absorbing more and more water very quickly. So as you heat the system, right, you build up and you increase the concentration. So somebody will come and tell me, okay, teacher, why are you telling us all this? This is outside of the book. No, it's not. You've learned osmotic pressure in your book. Your book says that osmosis is the most important phenomena in the physiological support system in the class. So now here I am, come to you in your final exam that will determine your career in the future. I'm going to ask you this simple question. If you know, so here I'm formulating a question for you. Okay, so I want you to think about it. If you know that osmotic pressure is calculated by this, let me clean up the system, right? I'm going to give you a question now as an examiner, if you know that the osmotic pressure can be calculated by this phenomena, and he tells you all that, so the examiner is going to give you all the facts, right? Based on your knowledge and understanding of osmosis, tell me, right, or draw for me, right, a histogram describing the relationship, right, between, or the difference in the two histograms between two different solutions, right, where the concentration of solution in one is 2% versus another which is 20%. And tell me what is the relationship between temperature, or how the osmotic pressure as a function of temperature. He's already telling you what this relationship is here. So your job now is to understand is to go back and remember your understanding for osmosis, right? And what it means really, the drivenness of water from one compartment to another based on this different concentration, and put that into this equation and see from this equation that you didn't know before, but now you know, right? The examiner is telling you that formula and is telling you what the definitions are. And he wants you to actually describe to him or to her the relationship between the temperature, for example, and pi. Draw a diagram showing this. Or is it this relationship? Is it this relationship? Is it this relationship? And he puts you on the y-axis pressure and time, right? And it could be any one of these other factors too. You could ask you any one of those number of things, right? And your job is to determine what's the right relationship. Is it like this? And it could be a multiple choice like that. Or it could be actually a long type of question where it's asking you to actually draw it for him, right? So you're going to have to draw it to him, right? So it will be something like this. As you increase the, as you move to higher temperature, right? You increase temperature, you increasing the pressure. You're increasing the temperature in this direction, you're increasing the pressure. Of course, up to a limit, because after that, you're reaching a limit where you're going to explode, right? So this is almost always the relationship in anything. You increase the concentration. Here's your true environment. You have very high concentration of the solute. You're going to keep increasing the pressure as you put more and more, but you're going to eventually saturate the system so that there's no more increase in that pressure anymore. And so you reach a plateau. So here's your curve. Same curve pretty much is going to apply to the molar concentration, the temperature, and this is constant, so this is no relationship, so this is a constant factor. And here's your question. Who helped us understand this? This guy. I know you all want to go to medical school. Most of you are now, by now, third year. You want to go to medical school. Some of you want to go to dentistry school or pharmaceutical school. But here's a man who dedicated his life to the study of gases and molecules and discovered a very important relationship that is now applied in many, 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 many different fields. That's why he won the Nobel Prize, because he, based on his studies and his scientific endeavor, he explained a physical phenomena in the universe that is very applicable to everything that we do in life. So if you haven't decided yet, right, what you're going to be doing in your career, what school you're going to go to, I want you to think about some of the greatest scientists, right, that have left behind them an impact that will affect humanity for eternity, so long as we live on this earth, right? How many doctors really have won Nobel Prize because of their impact on the field? Very, very, very few, right? But scientists, they spend their life studying these phenomena. Why? Because they see the beauty of it. They're thrilled, they're trickled by it. They don't sleep at night thinking about the problem. How can I explain that problem? Of course, they're going to have to invest a lot of education in getting to becoming these scientists, but I want you to actually think about it, right? Why would somebody just sit there and using all his time, right? Maybe it wasn't making much money, but at the time, maybe people in his neighborhood, could care less about this guy just going back here and there. He's not carrying his jacket and going to medical school and showing off. It's like he doesn't care. He's just like, he cares about something that really bothered him and he wants an explanation for it. Newton, same thing, right? Spent his life doing that. Einstein. These are people who are actually taking good education, right? Taking scientific methodology to come up with things that will impact us all in many different things. I mean, osmotic pressure right now is used in every single physical application, and that's just one of many greatest scientists, right? So I want you to think about it this way. You go to high school, you try to study something, try to love what you study, being physics, being chemistry, being biology, and if you don't make it to medical school, it's fine. It's fine. It's not the end of the world. Find something else. In fact, get a really, really good score and choose not to go to medical school because there's something equally important, even more important. Okay, so I really want to highlight this point on day one of class because I think science is very underappreciated, and our culture in the Arab world doesn't really value science in general because they just don't understand it. They haven't touched it. But I'm telling you, as a person who lives in the West, everything is driven by science. You're alive today because of the discoveries of biologists that are able to come up with a vaccine against coronavirus. If it wasn't for that, we would be all dead, right? It took us a few months to get to a vaccine, but here you go. It wasn't a doctor that discovered that. It was the labor and activities of many biologists. They had great training in chemistry and physics and in the sciences in general. And they eventually become great scientists, right? They have many applications. I'm not saying this is better or that's worse, doctor or not, but I'm saying that there are many things in life that are undervalued, and the sciences are one of them. And I want you to change your opinion about this topic. This is not a topic that you memorize and get good grades in school and go to medical school. This is a topic that's really important in its own right. Enjoy it, learn from it. Maybe that will be okay. Maybe you're going to become a biologist. Maybe you're going to become a geneticist. Maybe you're going to become the next Ahmed Zouel. We need more of those. We need some of those, okay? So that's that. I think I'm taking so long, but let me finish. I actually do have an example. I'm not going to go through it because of the small time, but here's an example again, right? So the steps, how you calculate it, you can measure the things that we talked about. You put them in numbers, right? And I'm, in fact, giving you an example, I think. So here's an example, right? Salt concentration. I want you to think about that. And I'm going to actually provide some quick answers and questions in the future, right at the end of this chapter, when we close the chapter, that will actually have used some of these things. So I want you to, when I upload that lecture, I want you to go through these numbers. And if I give you the number of, or the measure of the solution concentration, I'm asking you to calculate the osmotic pressure at a given temperature, because the osmosis or the osmotic pressure or any other pressure, because it's affected by temperature. So you must tell me you want the concentration or the percentage of the pressure value at what temperature, at what concentration, right? So you have to have each one of the elements of the equation defined so that you can give them the partial, the osmotic pressure in a given temperature, in a given concentration, in a given amount of the solution, right? At the end of the day, that value is measured in atmosphere. Unfortunately, I've seen many, some of the younger and even some of the older bio teachers using the wrong terminology, wrong measures as they introduce their lecture. And they say, oh, what if the osmotic pressure is 30%? Pressure is not measured in percentage. That's funny. This is like you asking me, how tall are you? And I'm going to tell you, well, I'm 120 kilogram. Excuse me, I'm asking you, how tall are you? Oh, I meant 300 kilometer. The unit is really important. You have to use the right unit. you cannot just say any numbers, right? And say percentage. Percentage means nothing in pressure unless you're talking about two solutions coming together and what's the relative pressure between one and another. Then in this case, you can use percentage. But pressure is measured in atmosphere. In atmosphere, right? So be careful, students and teachers alike. When you teach something, right? When you teach your student to use phrase, right? Pressure is not expressed in percentages. Pressure is not expressed as grams per mils. It's not expressed in kelvins. It's expressed in atmosphere or pascals, right? Or in bar or in millimeters of mercury if you're talking about gas or fluid, right? So it's a unit of pressure, right? It is commonly used to express atmospheric pressure. So I'm giving you some definitions here because unfortunately, students and even teachers alike, they're using the wrong terminology. And the reason why, right, I started giving some lectures in biology, both in Arabic and English, because I want to improve, right, and correct many of the falsehood in the material I introduced you. So teacher, if you are hearing me, please be careful for what measure are you using. Okay, don't tell me that I weigh 2 meters. I don't, right? I'm measured in either kilograms, right? Or in grams, or in micrograms, or in a ton, right? If you're big and things like that. So these are the right units. Don't use the wrong units to describe things that are not measured in these units. Students, be careful. Units are important, right? Atmosphere, liter, right? Because if I change any one of them, the entire equation collapses. You're talking wrong. You're not talking science. You're talking gibberish. You're talking garbage. You're talking trash, rubbish. You're not talking science. So talk science. Talk science. Okay, at the end, I did come up with a table for you. I want you to read these tables carefully. Again, as I mentioned, those of you that will follow me and join us in our group discussion on Facebook, I do want you to think about what I'm putting in this table, right? So what I'm trying to do here, actually I made this table myself, so is to highlight the differences between osmosis versus diffusion versus another concept that we didn't talk about, but you know of from your previous studies, right? And I'm highlighting what these differences are for you, right? So for example, I'm telling you that the medium is really important, right? So in this case, osmosis occurs across a semi-permeable membrane, right? We already spent half an hour discussing this important thing. Whereas diffusion, right, does not have a membrane in open medium, or occurs in material through a membrane, right, that is completely permeable. And I mentioned that already, right? If you have a membrane that's completely permeable, cell wall, right, osmosis doesn't exist. Things move in and out, water moves in and out by diffusion, not by osmosis, right? So that's the media, right? Or the medium singular, right? Saturation obviously also doesn't require a membrane, of course, in solutions, right? So then you can follow this table, the directions, right? From the high to the low. So we said that osmosis pushes things to move from pushes the water to move from the low concentration of water or high concentration of solutes, from the low concentration of the solutes or higher concentration of water to the medium in which there is a higher concentration of the solute or lower concentration of water. Different factors that affects it. That's why I described for you the Hof's equation, right? Van 't Hoff, right? Van 't Hoff equation, right? So for example, temperature also affects diffusion, right? So diffusion, let's say here. So temperature is a factor, right? You increase the temperature, you increase the ability of the different solute particles to move much quicker and therefore diffuse faster. Same thing for osmosis, same thing for this guy, right? So are there differences? Is there something else that affects that one? Direction, right? We talked about direction here in the case of diffusion. Obviously things move from the higher concentration to the lower concentration, irrespective of whether it's the solute or the solvent. I'm giving you some examples here. And then I'm also giving you another table that highlights the similarities, right? So this can come as a question to you at the end of the exam, right? So if I really want to formulate a very comprehensive question, I'll tell you, describe for me the process of osmosis. Tell me what kind of phenomena it is. Is it physical phenomena? I want you to think about that. Is it physical phenomena? Is it a biological phenomenon, right? Or is it a chemical phenomenon? And describe for me, how does it occur? What are the conditions required for it? And how does it differ from diffusion or saturation? I think if you go back to what you studied in last year in chapter 1, you pretty much will be able to answer that question, and the table simply helps you organize your thoughts. I think we'll stop here, and we'll continue to talk about the support system and movement. We'll focus more on the movement and the structural elements of the support system in the second lecture. I hope you had fun following me. If you have any suggestions, and this is the very first lecture for me in giving you a lecture in English to people who live in Egypt, learning sciences in English. This may be a little bit new experience to you and maybe to me, certainly to me, but maybe to you as well. And so tell me if there's something that you want me to do, to adapt in these lectures, so that, for example, if you didn't understand a word from what I said because I spoke mostly in English, tell me that, in which case I'll go back and exchange things between English and Arabic, maybe more in Arabic, so that you follow me freely. So I'm looking forward to having your feedback on our group. If you haven't subscribed to our YouTube channel, please do so. If you haven't signed up for our groups on Facebook, please do so, so that you can have access to that material. And I hope you had enjoyed that hour and a half, 2 hours. And we'll see you next time. Good luck. Bye-bye.